Hello, and welcome to your 4th week of neuroscience tutorial videos. This week we'll be covering eyes and ears. I am Tommy Moran, and I've also taken over for your neuroscience coordinator Jono in drawing today, so you'll have to bear with me in that regard. Today we'll be covering 3 main topics: the retina and the neural circuits that run from the retina. We'll be covering coordination of eye movements, and then we'll finish up with perception and localization of sound. So, moving right along, I first wanted to take a look at the structures that light has to pass through in order to get to the retina. At the front here we have our cornea, and deep to that the aqueous humor. Behind that is our iris, and then our lens, which is supported by our zonules and our ciliary muscles. Posterior to that, in the posterior chamber, is the vitreous humor. So these are the main optic factors that light has to pass through to get to the retina. I've also labeled here the choroid, which is the vasculature supply to the retina, and the sclera, which is the connective tissue outside supporting it. This small indentation here is the fovea, which is a collection of cones that allows for advanced acuity in the perception of light. We're going to take in a small zoomed-in section of the retina here and analyze the cells that compose the initiation of this neural circuit. The retina is composed of predominantly 3 nuclear layers. We have our photoreceptor cells, our bipolar cells, and our ganglion cells. The photoreceptors are located in the outermost nuclear layer, which is in respect to the center of the eye, and that's why it's referred to as the outermost. Our bipolar cells are then in our inner nuclear layer, and our ganglion cells in our innermost nuclear layer. You'll notice here I've also drawn the horizontal cells, which is a connection between the photoreceptors and bipolar cells. These are considered in the outer plexiform layer, and we have our amacrine cells in our inner plexiform layer, providing more connections between the bipolar cells and the ganglion cells. So if we look back at our eye here for a moment, we can notice how light has to travel through all the optic structures and then also through the ganglion and bipolar cells before it can actually reach the photoreceptor cells and the opsin within those photoreceptor cells to initiate transduction of light. The one exception here being in the fovea, where our ganglion and bipolar cells are folded out of the way, again to help improve the visual acuity of the fovea. So let's take a look now at how the photoreceptor cells are actually able to respond to the signal of light and initiate the depolarization of the ganglion cells to send a signal to our primary visual cortex. I've drawn a photoreceptor cell here, and you'll notice I've drawn light binding to an opsin. When our photon, which is light, binds to this opsin, it causes a conformational change such that the opsin is able to activate a G protein that's associated with it, such that that G protein may bind GTP and itself then activate a phosphodiesterase. Phosphodiesterase will break down cyclic GMP into GMP. The result is a decreased concentration of cyclic GMP. And what this causes is the closure of cyclic GMP-gated sodium channels. Effectively, by closing these sodium channels, we cause the photoreceptor cell to hyperpolarize. And in its hyperpolarized state, it isn't able to release intracellular calcium stores that are necessary for the fusion and release of the neurosecretory vesicles. Within these vesicles would usually be glutamate. So what we see is when light binds to the photoreceptor cells, we release less glutamate. Now, how does this transmit a signal to the ganglion cells? It does so through one of 2 different parallel pathways, the on bipolar cell or the off bipolar cell. These 2 pathways are effectively a redundant mechanism that has evolved sheerly because of the importance of being able to distinguish signals from light. If we look first at our on-bipolar cell, we see it has a metatropic receptor, which would usually respond to glutamate and activate an inhibitory or a GI protein, which would inhibit the bipolar cell from depolarizing and sending a signal to the ganglion cell. Consequently, in the absence of glutamate, the cell is able to depolarize and propagate that action potential. Conversely, our off-bipolar cell has an ionotropic receptor, which means that when it is bound to glutamate, it opens a sodium receptor. The opening of this sodium receptor in the presence of glutamate would usually allow the off-bipolar cell to depolarize. However, in its absence, this ionotropic receptor is closed, so the bipolar cell remains hyperpolarized. In this way, the on-bipolar cell is able to translate the signal that light is present in the photoreceptor, and the off-bipolar cell remains inactive. However, if we take away light and we walk through this pathway, we'll see we no longer activate the associated G protein with our opsin, which means that we don't activate phosphodiesterase, which means we don't break down cyclic GMP, and our cyclic GMP-gated sodium channel remains opened, which causes depolarization of the photoreceptor cell. We'll then see release of our intracellular calcium stores, which facilitates the fusion and the release of that glutamate. Now, when we look at the off-bipolar cell, glutamate will bind to the ionotropic receptor and allow depolarization of the bipolar cell. However, as I mentioned earlier, the metabotropic protein on the on-bipolar cell is bound to an inhibitory G protein, so our on-bipolar pathway will shut off. This effectively sends a signal to the visual cortex saying that the photoreceptor cell is in the dark and isn't seeing light. The last concept I want to draw attention to in this complex network of cells in our retina is the center surround effect. In the diagram we drew earlier, we have a collection of photon receptors with connections through direct and indirect pathways to the blue ganglion cell I've drawn here. This center through pathway and the two adjacent indirect pathways constitute the receptive field of that ganglion cell. The through pathway is stimulatory and leads to depolarization and the propagation of a signal to the visual cortex. However, the photoreceptors that communicate through the horizontal cells are inhibitory because horizontal cells secrete GABA and thus reduce the activity of the ganglion cells. This creates the center surround effect of its receptive field, which helps us to distinguish discrete edges in our vision and improves contrast. Now we'll quickly cover the pathway of light from the retina to the visual cortex. The primary visual cortex is in the occipital lobe with the superior and inferior bank separated by the calcarine fissure in both hemispheres. Importantly, the left retina receives the right visual field and vice versa. From the retina, both signals travel through the optic nerve to the optic chiasm, where we see both of the nasal portions decussate and thus separates our field of vision. Tracing the right visual field here from the left optic tract to the left LGN and through the optic radiations, we can trace the pathway back to the primary visual cortex. Patterns in various visual field defects help us to discern the location of a particular lesion when working up a patient. For instance, a defect of a right inopia, such as this, would be an injury to the optic nerve, the retina, or to the orbit itself. A bitemporal hemianopia is a pathognomonic sign of damage to the optic chiasm, which is commonly caused by a pituitary adenoma or an aneurysm of the anterior communicating artery in the circle of Willis. Damage to the optic tract causes a homonymous hemianopia in the opposite side's field of vision, and damage to the optic radiation towards the inferior bank causes a quadratic anopia of the upper field of vision. Finally, a homonymous hemianopia that has macular sparing occurs from damage to the occipital lobe from a posterior cerebral artery infarction. because the macula, which is located centrally and posteriorly in the visual cortex, has collateral supply from the middle cerebral artery. You can see how using these visual defects is clinically quite significant in order to help determine the location of a lesion. We can also use the pupillary reflex in a similar manner. An interesting function of some ganglion cells in our innermost nuclear layer of the retina is the capacity to respond to light. These are called intrinsically photosensitive ganglion cells. They have opsins that are able to bind light and stimulate a neural signal without the activity of photoreceptors. This afferent axon travels down the optic nerve and the optic tract, leaving the rest of the axons just before the LGN, traveling to a pretectal nucleus. You can see I've drawn here just interior to the tectum in the midbrain. This pretectal nucleus then relays a signal to the Edinger-Westphal nucleus, which you'll recall is the parasympathetic nucleus of the oculomotor nerve. The reflex then immediately sends an afferent signal back out with the rest of the axons in the oculomotor nerve to the ciliary ganglion, which then goes on to cause constriction of our pupil, thus completing our pupillary constriction reflex to light. However, back at the pretectal nucleus, we also send an additional axon or interneuron across the midline to the contralateral Edinger-Westphal nucleus, which then follows the contralateral oculomotor nerve and causes constriction of the opposite pupil. This is called the consensual reflex. So in response to light, both of our pupils will constrict simultaneously. The function of the consensual response clinically allows us to assess the functionality of the efferent nerve of that eye. By shining light into the left eye as we have here, we bypass the afferent pathway of the right eye. Thus, if there is an afferent defect of the right eye, although we might not see constriction when we shine light directly into the right eye, we should still see it when light is shone into the left eye, telling us that there is an afferent defect of that right eye. This completes our first objective, reviewing the retina and the neural circuits projected from the retina. Next, we'll move on to the coordination of eye movements. And to do this, I'd like to use the example of the vestibuloocular reflex. I've drawn here two eyes, a left eye and a right eye, both with a lateral rectus and a medial rectus. And at the back, I've drawn a horizontal semicircular canal of the left ear. Now, our vestibuloocular reflex functions to help keep our eyes fixed on a point whenever our head turns. So if our head turns to the left, as we'll draw in this example, our eyes would have to turn right. And all you have to do throughout this example is keep in mind the endpoint, or our objective, and what muscles would have to contract or relax to make that happen. So from the vestibuloocular nucleus, we send first two projections on the left side, keeping in mind the objective is inhibit the left lateral abducens, or left lateral rectus, and activate the left medial oculomotor, or the left medial rectus. It thus sends an inhibitory signal to the abducens and a stimulatory signal to the oculomotor, and that takes care of our left eye entirely. From this point on, all we have to consider is the right eye. Now, bear in mind that throughout this example, we would also have reciprocal input from the right vestibular nerve. Keeping in mind the objective again, we want to see activation of the right abducens this time, and inhibition of the right oculomotor, just the opposite. The left vestibular nucleus will give off a stimulatory signal to the right oculomotor nucleus, which takes care of that. But what we will also see is that the abducens nucleus, on either side, has the capacity to give off a positive or negative signal depending on its own activity. So in this example, since our left abducens is inhibitory, it will inhibit the right oculomotor, and the same thing occurs vice versa. These connections between cranial nerve nuclei are called our medial longitudinal fasciculus, and produce an interesting presentation when damaged called internuclear ophthalmoplegia, where we lose the ability to contract both of the medial rectus muscles. Finally, we'll move on to our third objective, the perception and localization of sound. Sound comes in from the outer ear and transmits physical energy through the tympanic membrane into the ossicles, which is then relayed into physical energy onto the non-compressible perilymph through the oval window. Generation of waves transmitted through the scala vestibule and transmitted around the cochlea through the scala tympani is transmitted into the inner compartment, the scala media, where our high potassium concentration facilitates activation of hair cells in the organ of Corti in conjunction with the frictional forces between the basilar and tectoral membranes. This stimulates release of neurotransmitters, which is propagated through our afferent cochlear nerve. I've also drawn here a stretched out version of the basilar membrane just below to illustrate the fact that higher frequency sounds, which have shorter wavelengths, are registered by the base or the proximal portion in the cochlea, and the apex recognizes the lower frequency and longer wavelength sounds. Moving on to localization of sound now. There are effectively two mechanisms by which we are able to relay the signal to our brain of where things are perceived in space when we hear them. We segregate low frequency and high frequency sounds. The perception of our low frequency sounds in the space around us utilizes the timing of different sounds and is transmitted to the medial superior olive. In the diagram I've drawn here, there's a sound being transmitted from the right side of our head. This sound will thus reach our right ear significantly before it reaches our left ear. So the signal that gets relayed to our auditory cortex will be able to transmit that delay in time and say that the sound we're hearing, this low frequency sound, must be coming from our right ear. In contrast, our high frequency sounds utilize the loudness of a sound in order to localize it in space. In a similar diagram here, with sound coming from our right ear again, you can see that sound as it hits our head creates a shadow and thus becomes quieter by the time it reaches our left ear. High frequency sounds are relayed to the lateral superior olive, and the one that registers the louder intensity sends an inhibitory signal to the opposite. Thus, once these signals are relayed to the auditory cortex, the one that has the stronger signal, in our example here being the right, is able to translate the location of that sound in space. That completes our third objective of the day. Thank you everyone for joining me again this week, and we'll see you next time for week 5. Cheerios!